structure and they are competing for the active site. And if they are competing for a single active site, means only one thing can bind at the active site of the enzyme. Either your inhibitor will bind or your substrate. Either your inhibitor will be binded with it or your substrate will get binded to it. Any one thing will bind with it, right? Either your inhibitor or your substrate will bind. Here you can see. So for any kind of inhibition, what you need to know is whether your Michaelis Menten constant is changing, Vmax is changing. These two things you need to know, right? So Michaelis Menten constant is also called as Km value. So what will be the effect on Km value and what will be the effect on Vmax value? These two things will always be kept in mind while reading the inhibitions. So first one is competitive, and let us see how the value changes. As we know that this is our Michaelis Menten graph. This red one is without inhibitor and this black one is with inhibitor. So without inhibitor and with inhibitor. So what's happening without inhibitor? It's a normal hyperbola, right? And here it's quite straight. Means there is some change in the Vmax value and Km value, right? So Vmax value and Km value. Here you can see the both of the hyperbolas, they are touching the same place on the y-axis. That means that Vmax is remaining unchanged. And here half of the maximum velocity when it is attained this place, half of the maximum velocity when it is attained, the graph where it is cutting on the x-axis, that is the Km value. So in case of with inhibitor, it's cutting at somewhere different place than the Km. So here it's like towards the positive. So Km value is increasing. So what can be said is that Km value increases in case of competitive inhibition and Vmax remains unchanged, right? Km value increases and Vmax value remains unchanged. And your inhibitor and substrate, they both will bind at the same place in the enzyme, that is the active site. Thus, it is also called as competitive inhibition, right? Let's move. So what are the features of competitive inhibition? First of all, if they both are competing for the same place, it means they are structural analog. They both have the same structure. That's why they can bind at the active site of enzyme, right? Then another thing, whenever we are talking about inhibition, whether it's reversible or whether it's irreversible, this is also one of the factor. So when it is competitive inhibition, it is reversible. So what are the factors which will affect the reversibility of the reaction? How it will be reversible? If in the reaction mixture there is an enzyme and there is inhibitor and there is substrate also. So this inhibitor and substrate, they are competing for the enzyme. But now if we increase the concentration of substrate to a higher level, higher amount, what will happen? The enzyme will encounter most of the time substrate, and thus it will give to a positive reaction. Thus, it can be reversed when excess of the substrate is added. Excess substrate abolishes the inhibition. Enzyme and inhibitor and substrate in the environment, if they are present and there is lots of substrate, means your substrate will come in contact with the enzyme and it will affect the reaction positively. Inhibitor won't get any chance to interact with the active site of the enzyme. They are structural analog and they are, and if the concentration of substrate is increased, the reaction will proceed in the positive direction. Vmax remains the same. The value of Vmax remains the same. There will be no effect on the Vmax value and Km increase. So what is this Km? What does this Km actually tells us about? what you can infer from Km, or if Km is coming in your mind, what all things should come in your mind. Km tells us that it's a Michaelis Menten constant, first thing. Then what about Km? It is that substrate concentration where half of the maximum velocity is attained. And what does it tell us about the reactivity? When Km value is low, it means that substrate is having higher affinity for the enzyme. The value of K is higher, it means that it will have lower affinity towards enzyme. So it tells us about the affinity. If Km is higher, it means low affinity. If Km is lower, it means higher affinity. So whenever Km you are coming across Km, you should think about affinity. And another thing that you should keep in mind is Km is inversely proportional to affinity. Though this is not a formula, but this you should keep in mind so that you can just solve the questions. Because most of the time questions will be asked that if Km value is higher, how will it affect the affinity? If Km value is lower, how it affects the affinity? So just remember that Km is inversely proportional to affinity. So you'll be able to solve the questions, because many questions which are based on enzyme inhibition, they are almost direct questions, right? Let's move. So here in this table, I have included some of the enzymes which are inhibited by the drugs. So the enzymes which are inhibited by the drugs, we'll just revise the names and we'll be seeing the mechanism of one or two of them. Right? Vitamin K epoxide is inhibited by dicomerol. Dihydrofolate reductase is inhibited by methotrexate. Acetylcholine esterase is inhibited by succinylcholine. So these are the enzymes which are inhibited by these drugs, so they can be helpful. Now there are some of the enzymes which are not inhibited by drugs, which are inhibited by some other some other kind of competitive inhibitor. So in the next table, I have highlighted the name of the enzymes and the substrate which binds with the enzyme in the normal reaction, and then the competitor which affects the binding of enzyme with the substrate, right? So the enzyme lactate dehydrogenase, it acts on the substrate lactate, and the competitive inhibitor is oxamate. Aconitase enzyme is inhibited by transaconitase, and its normal substrate is cis-aconitate. Succinate dehydrogenase, competitive inhibitor malonate, and its normal substrate is succinate. HMG-CoA, its competitive inhibitor is HMG, and its competitive inhibitor is HMG, and the substrate which it reacts with in a normal reaction, HMG-CoA. So these are some of the examples. You should keep all these examples in your mind because questions are asked on these examples or these combinations. Now coming to the next inhibition, that is non-competitive inhibition. So as the word itself is saying that there will be no competition between the substrate and inhibitor for binding with the enzyme. Enzyme will have a separate binding site for inhibitor and for substrate. So how we can draw this? If this is your enzyme and this is the binding place for your substrate, this place is for the inhibitor and this is your enzyme. It can bind with inhibitor at another place and it can bind with substrate at another place. So this is the kind of non-competitive inhibition. There is no competition. They both can bind at different places. Then they both can bind together and they both can be involved in the reaction. So as I told you, what all things we need to see when we are talking about inhibition, we need to see the Km value, we need to see the Vmax value, and we need to see the reversibility of the reaction. So here in this case, what is happening is, what first of all, reversibility. So reversible non-competitive inhibition are very few and mostly they are irreversible. They are irreversible in nature mostly, and there are very few which will be reversible. right? So the structure of substrate and inhibitor, they are not analogs. They are not similar in structure. So by increasing the concentration of substrate, the reaction cannot move in the positive direction, or we cannot stop our enzyme to bind with the inhibitor if there is lots of substrate. It will either way bind with the inhibitor, whether the concentration of substrate is higher or it's not higher. So how your Michaelis Menten graph changes. This red line tells about in absence of inhibitor, and this black line tells about in presence of inhibitor. So here you can see that the without inhibitor graph is touching somewhere here on the y-axis, and with inhibitor it is touching over here. It means there will be depression in the Vmax value. Vmax will decrease when there will be inhibitor in the reaction. And if we draw a line from the half of the maximum velocity in presence of inhibitor and in absence of inhibitor on the x-axis, what we can see is they both are cutting at the same place. It means there will be no effect on the Km value. So what about the reversibility of the reaction? What about the reversibility? In most of the cases, it is irreversible. Irreversible. And how the Vmax and Km changes? Vmax decreases and Km value remains unchanged, and it is also directly linked with no matter how much you add the substrate, no matter you increase the concentration of substrate, there will be no effect on the concentration of the there will be no effect on the course of the reaction. The enzyme, if it has to bind with inhibitor, it will bind. If it is binded with inhibitor, then it won't be binding with substrate, but if it is bounded with substrate, it can bind with the inhibitor. This is the condition for non-competitive inhibition. So what all are possible? Enzyme can bind with substrate, enzyme can bind with inhibitor, and if enzyme has bound with the substrate, it can bind with inhibitor also. These three conditions are there for the non-competitive inhibition. And remember that no matter how much substrate you increase, there will be no effect on the binding of enzyme with the substrate. Then it's irreversible in most of the cases. Vmax is decreasing and Km value is remaining unchanged. Right? So what are the features of non-competitive inhibition? What are the features of non-competitive inhibition? Inhibitor have no structural resemblance to the substrate. They are not similar in structure at all. There is no resemblance. Mostly irreversible. Excess of substrate do not abolish the inhibition. Km value remains the same and Vmax decreases. This we have seen in the last slide also. Now some of the example and along with the mechanism. The non-competitive inhibition, here you can see the inhibitor and the name of the enzyme. So cytochrome C oxidase is inhibited by cyanide. Glyceraldehyde 3-phosphate is inhibited by iodoacetate. Enolase is inhibited by fluoride. Acetaldehyde dehydrogenase is inhibited by disulfiram. SH groups of various enzymes are inhibited by dimercaprol. Alpha-ketoglutarate dehydrogenase is inhibited by arsenite. Aconitase enzyme is inhibited by fluoroacetate. Serine proteases are inhibited by diisopropyl fluorophosphate. So these are some of the examples. You should remember these examples. I am explaining the mechanism of one of the example. That is how the fluoride, it inhibits the enolase enzyme. I am going to explain you. So whenever there is a test for determination of plasma glucose, estimation, we can say. So what happens is a gray bulb is used. So what is there in this gray bulb? What is the composition of the gray bulb? It is also called as fluoride bulb. It is also called as fluoride bulb. And what is the composition of fluoride bulb? It contains sodium fluoride and potassium oxalate. These two are the things in the fluoride bulb: sodium fluoride and potassium oxalate. So now it inhibits the enzyme enolase. We are saying that it inhibits the enzyme enolase. So where does it binds this enolase, and which combination binds this enolase is fluoride, P and magnesium. Potassium, fluoride, potassium, magnesium, it binds enolase and it binds it in the irreversible manner. And as soon as it binds the enolase, it will stop the glycolysis pathway, will stop glycolysis. So if glycolysis is inhibited at the time of extraction of blood, the glucose level won't go down. So this is how the plasma glucose estimation performed. And the tube, it contains a gray bulb. And what is the composition? Sodium fluoride and potassium oxalate, and it inhibits the enzyme enolase, right? You can remember this thing, and there are mechanisms for each and every inhibitor which inhibits the enzyme and the reaction which it is inhibiting, that we can see in some other class, and we can see how it happens. Then comes a suicide inhibition. So what is this suicide inhibition? What happens is, if your enzyme, it is present in an environment in a normal way, and there is an inhibitor also, so they won't be reacting much, they won't be bothering each other much. But if this inhibitor, it comes in contact with the active site of the enzyme, this normal inhibitor will change into a deadly inhibitor, that otherwise a normal inhibitor, which is okay to inhibit the enzyme activity, now it is deadly, will be binding with the enzyme and it won't be leaving the enzyme at all, will be binded with the enzyme irreversibly. So this is a kind of irreversible inhibition, and another name given to it is mechanism based inactivation. What's the name? That is mechanism based inactivation, and it's a special class of irreversible inhibition. So inhibitors are relatively unreactive when we talk about suicide inhibition, until they bind with the active site of a specific enzyme, until they come across the specific site of enzyme, that is the active site. And once the inhibitor binds to the enzyme by the action of enzyme, it is converted to a potent inhibitor. Otherwise, it's a non-potent inhibitor if it is lying in the environment. But when it binds with the enzyme, it changes its shape and it converts into a potent or a deadly inhibitor. Right? So some of the examples of suicide inhibition, like three examples I have included over here based on the exams they have been asked. The first one is allopurinol. It inhibits xanthine oxidase. Allopurinol, it inhibits xanthine oxidase. Allopurinol converted to alloxanthine, which irreversibly inhibit the enzyme. Allopurinol is a normal inhibitor, but alloxanthine is a deadly inhibitor or a potent inhibitor. So when allopurinol is converted into alloxanthine in presence of the enzyme xanthine oxidase, it gets converted into a deadlier form and it binds with the enzyme irreversibly. Then another thing, for the treatment of trypanosomiasis by difluoromethyl-ornithine, what does it do? It inhibits the enzyme ornithine decarboxylase. Inhibits ornithine decarboxylase, and the disease that is being treated is trypanosomiasis by difluoromethyl-ornithine. Third example, keep in mind because this was asked in one of the examination. This is a PYQ. Directly it was asked. Aspirin acetylates the active site of cyclooxygenase and thus inhibits prostaglandin synthesis. So aspirin, it acetylates the cyclooxygenase and inhibits the synthesis of prostaglandin. Aspirin is performing the suicide inhibition of cyclooxygenase, and cyclooxygenase is responsible for synthesis of prostaglandins. Thus, these are the examples of suicide inhibition. First one was your allopurinol, its deadlier form is alloxanthine. Then for the treatment of trypanosomiasis, it inhibits the enzyme ornithine decarboxylase by difluoromethyl ornithine, and aspirin, it acetylates the active site of cyclooxygenase and thus there will be no production of prostaglandins, right? Let's move. Now this is the last kind of inhibition, and before moving to this, let us have a look at uncompetitive also. Very rarely questions are asked on the basis of uncompetitive. Uncompetitive inhibition. So what is this uncompetitive inhibition? What you need to remember is the Vmax value and Km value for uncompetitive inhibition. So Vmax value decreases and Km value decreases in case of uncompetitive inhibition. So we have seen competitive, we have seen non-competitive, and we are seeing uncompetitive. So don't get confused between non and uncompetitive. They both are different things. Uncompetitive and non-competitive. The condition for uncompetitive and non-competitive is different. For uncompetitive, what we were saying is Vmax decreases and Km value remains unchanged. But for uncompetitive, what we are saying is Vmax value goes down and Km value also goes down. So don't get confused between uncompetitive inhibition and non-competitive inhibition. They both are different things. Uncompetitive inhibition is a very rare kind of inhibition and hardly any questions are asked, but I have told you if there is a question, just remember Vmax value and Km value, both of them decreases. Now feedback inhibition. What is this feedback inhibition? It is also called as end product inhibition. Also called as end product inhibition. Feedback. So what is the last product being formed will inhibit the first reaction. First reaction, X is getting converted into Z by first enzyme, then Z is converted into A, and this A is converted into B. So it is inhibiting the first reaction, feedback inhibition or loop inhibition. The end product will inhibit the first reaction. Why? Because if end product is formed in the required amount in the environment, why would it let the formation of more end product? Why? Because it is already there, it's not needed now anymore. So why unnecessary production? So feedback inhibition and product inhibition. Example which you should keep in mind is the activity of enzyme inhibited by the final product of the pathway. One of the example is AMP. If it is formed in the environment, it will inhibit the formation of further purines in the environment. So a quick PYQ we can do over here is, if we are saying that production of AMP inhibits the first step in purine synthesis. So this purine synthesis, what are the purines in our body? We say A and G. These two are the examples, or these two are the bases we talk about, right? But when we are talking about purine synthesis, which nucleotide is formed first? It is IMP, inosine monophosphate. Many a times you people will get confused between AMP or GMP. But never forget IMP. What is most important? IMP, inosine monophosphate. So in purine synthesis, what thing is formed first? IMP that is formed first. So just remember IMP. It's not AMP, it's not GMP. Though inosine monophosphate is less common purine, but is the first purine that is synthesized in the purine synthesis pathway. And it was a very important PYQ. It was asked in 2-3 examination, right? So here what we were saying is if there is lots of AMP in the environment, will inhibit further formation of more purine in the environment. More purine won't be formed, right? So this is end product inhibition or feedback inhibition. Let's move further. Here I have included some of the PYQs and I have also given the answers over here. I'll be explaining each question and we'll be solving them so that you'll have an idea that how the questions are asked on enzyme inhibition, right? So the first question is the type of enzyme inhibition in which succinate dehydrogenase reaction is inhibited by malonate. So I told you while we were doing that table, I told you just remember the table. So I've gone through that table nicely and you have learnt all of the things, you will be able to do the question. So it is a kind of competitive inhibition. Enzyme inhibition in which succinate dehydrogenase. Your enzyme is succinate dehydrogenase. It is getting inhibited by malonate. So this is an example of competitive inhibition, right? So try to remember that table well and you will be able to solve some of the questions because they are directly asked on the thing. Coming to the next question. Features of competitive inhibitions are. So we know about competitive inhibitions. We have seen three major features about the reversibility and irreversibility. So it is a reversible kind of reaction when there will be high substrate concentration. Then how the Vmax and Km changes. So Vmax remains unchanged and your Km value it increases. That is the affinity decrease. So what are the conditions over here which is fulfilling these things? Vmax increases. So no, there is no effect on the Vmax. Km increases. So yes, the Km value increases. Vmax decreases. So again, there is no effect on the Vmax value. Then Km decreases. No, we have said that Km increases. And Vmax remains constant. So yes, Vmax remains constant. There is no effect on Vmax value. So these are the features of competitive inhibition. Now coming to the next, which is true about enzyme kinetics for competitive inhibition. What is true about enzyme kinetics? So let us see what is true. In competitive inhibition, low Km, high affinity. So no, we know that Km value is higher. So high Km, high affinity. So yes, true. High, affinity, it decreases. So high Km, low affinity. Yes, true. Low Km, low affinity, no. Right? So if the value of Km is If the value of Km is higher, it means there is low affinity of your enzyme with your substrate. This is the condition for competitive inhibition. Let's move to the next question. Non-competitive enzyme inhibition leads to. What does it leads to in non-competitive inhibition? Vmax value, we know that the graph changes like this. So there is a change in this is without inhibitor and this is in presence of inhibitor. So what happens to the Vmax value? Vmax value decreases or the Vmax decreases. And for competitive, there is no change in the Vmax. Here, the value of Vmax is decreased. Let's move to the next question. Non-competitive reversible inhibitors. What does they do? Raise Km value? No. There is no effect on the Km. Lowers the Km value? No, there is no effect. Lowers the Vmax. So yes, in just last slide we have seen that it lowers the Vmax. Raises both Vmax and Km. So no, possible. do not affect either Vmax or Km. No, this is also not the condition. The condition is it lowers the Vmax and there is no effect on the Km value. True about competitive antagonism. What is true? Vmax increase in competitive inhibition. No, there is no effect on Vmax. They are substrate analog. So yes, they fight for the same place and they bind at the same place in the enzyme, that is the active site. So yes, they are the substrate analogs. Reversible reactions. So yes, they are falling into the category of reversible reaction. Then Km increased. So the value of Km increases and affinity decreases. This is true. Vmax decreased. No, there is no effect on the Vmax. The answer is B, C, and D. So this is what is true about competitive antagonism. Then next question. Km changes and Vmax remains the same. Km changes and Vmax remains same. What is the type of enzyme inhibition? I've seen this condition many a times, and these are some of the very favorite questions of the examiners and ask it every time. So the answer is competitive inhibition directly. Now we have learned this thing many a time. So the answer is competitive inhibition. Non-competitive inhibition, no. In non-competitive, conditions are different. Value of Km remains same. Max decreases. So here you can see the word uncompetitive, which I was telling, which is quite rare. So uncompetitive and non-competitive, they both are different. What is the condition for uncompetitive? Both of the values for Km as well as Vmax, they both decrease. Suicide inhibition, it is altogether a different category of inhibition. So here the answer is competitive inhibition, because only in competitive inhibition Km value and Vmax value, Km changes and Vmax remains same. So this is it for today's class, and we'll be seeing some more interesting lectures in the upcoming classes. Important topics and interesting topics we can discuss in the upcoming classes. You can just write down in the comment box which topics you want to learn in biochemistry, and I'll be teaching you those topics in biochemistry. And lastly, if you like my video, please don't forget to like the video, and for getting the notifications for all of the upcoming videos, just hit the bell icon, and also don't forget to subscribe. Let's crack NEET PG. And lastly, if you're going for the plus subscription, don't forget to use my referral code, that is FB01. So by using my referral code, you will be getting an additional 10% discount. So please like the video, and whatever topic you want to read in biochemistry, just write in the comment box, because I will check that comment and we can just read that topic. Okay, that is it for today's class. Happy learning. Hope you enjoyed the topic which I covered today. And yeah, bye. Good night. Happy learning.